Today's topic of discussion is about recombinant DNA and biotechnology. First of all, let us have a basic idea of what exactly is recombinant DNA technology. In this technology, DNA molecules are extracted from different sources and then they are chemically joined together. So basically, two different DNA are being joined together. For example, DNA comprising of an animal gene may be recombined with the DNA from a bacterium. Animal gene and bacterial gene are extracted and joined together. Now let us have a look on discovery of recombinant DNA technology. First of all, structure of DNA was discovered by Watson and Crick in 1953 and rDNA technology was discovered by Paul Berg in 1972. First plasmid vector was discovered by Cohen and Boyer in 1973. Coming to the goals of recombinant DNA technology, primary goal is to isolate and characterize a gene and to make a desired alteration in one or more isolated genes. Another goal is to artificially synthesize a new gene and also alternating the genome of an organism. And also rDNA is used in understanding the hereditary disease and their cure. Now let us discuss about procedure of making the rDNA. Procedure of making rDNA involves 4 different steps. First step is to isolate the DNA. Second step is to cut the DNA. Third step is to join the DNA, finally amplifying of the DNA. Let us now discuss about each and every step in detail. First of all, isolating of the DNA. In order to isolate the DNA from the cell, first of all the cells should be treated with some enzymes called lysozyme or the cellulase to break down the cell wall. After that, they are treated with denaturing agents which will disrupt the cell membrane as well as the proteins. Then they are treated with the enzyme protease which destroys the proteins RNA and RNase. So till now, cell membrane, proteins, RNA and RNase are disrupted. Now centrifugation is done. Centrifuge is used to separate the cell debris from the supernatant which contains the DNA. Because all the cell components are disrupted and only the DNA is left out, supernatant contains DNA. Then it is treated with ethanol which precipitates the DNA to form the viscous strands. Finally, we are isolating the DNA. After isolating, second step is to cut the DNA. DNA can be cut into large fragments by mechanical shearing. One particular enzyme called restriction enzymes are the seizers of the molecular genetics. Let us now know about this important enzyme called as restriction enzyme. So these restriction enzymes are the special class of sequence-specific enzymes found in bacteria. And restriction enzymes are site-specific, that means they cut the DNA at specific site and cleave the DNA molecules only at specific nucleotide sequence. Restriction endonuclease recognize the DNA at palindrome sequence. Palindrome is nothing but a nucleotide sequence reading same forward and backward. So restriction enzymes cut the DNA at only the palindrome sequence. And also restriction enzymes make staggered cuts with complementary base sequences for easy attachment. If we can see in this picture, this particular site is a palindrome sequence. So restriction enzymes cut the DNA at this particular site. Palindrome sequence means nucleotide sequence GAATTC reading same forward and GAATTC reading same backward. So at this particular site, the restriction enzymes come, bind and cut the DNA. After cutting the DNA, the sticky ends are left out so that it will be easy to join the two DNA segments later in joining of the DNA. So after cutting the DNA, let us talk about joining of the DNA. If you can see in this picture, after cutting of the DNA, two sticky ends are left out. So these two sticky ends should be binded by one enzyme. What is the enzyme present here? It is DNA ligase. DNA ligase joins the two cut DNA segments and finally forms the recombinant DNA. After joining of the DNA, final step is to amplify the DNA. Amplify means multiply. So multiplying the recombinant DNA. How it is done? It is done by transforming the recombinant DNA into the bacterial host strain. So let us know the procedure. First of all, the bacterial cells are treated with the calcium chloride. After that, the recombinant DNA is added. Then the cells are heat shocked at 42 degrees Celsius. Then the recombinant DNA enters the bacterial cell by an unknown mechanism. So finally the recombinant DNA has entered the bacterial cell. Then the recombinant DNA will be replicated in the bacterial cell. So whenever the bacterial cell divides, the replicated recombinant DNA molecule also goes to the daughter cells. So each and every daughter cell is having the recombinant DNA. That means the recombinant DNA is multiplied. Thus DNA is amplified. That means multiplied. Firstly, DNA was isolated, followed by cutting of the DNA, then joining of the DNA, finally amplifying the DNA. If you can see in this picture, this is a recombinant DNA molecule and this is a bacterial chromosome. And recombinant DNA molecule is introduced into the bacterial cell. Then what happens is whenever the bacterial cell is dividing, the recombinant DNA molecule enters all the daughter cells. So as the bacterial cell divides, recombinant DNA molecule keeps on entering the daughter cells. So this is how the recombinant DNA is being amplified. So that was all about the procedure of rDNA. Now let us discuss about the enzymes used in recombinant DNA technology. First enzyme is DNA ligase. It is used to bind the DNA molecules. And the other enzyme is reverse transcriptase. It makes a DNA copy from an RNA molecule. And exonuclease 3 removes the nucleotide residues from the 3' end. Bacteriophage exonuclease removes the nucleotides from the 5' end. Alkaline phosphatase removes the terminal phosphates. So each enzyme has got its own property. Now let us discuss about vectors used in rDNA technology. What is a vector? Vector is nothing but a DNA molecule used as a vehicle to artificially carry the foreign genetic material into the another cell, where it can be replicated or expressed. So vector is nothing but a DNA molecule. So vector is an area of a DNA that can join another DNA part without losing the limit of self-replication. So DNA doesn't lose its property. So vectors should have selectable marker to indicate which host cells receive the recombinant DNA molecule. So the selectable marker is very helpful in evaluating which host cell has received the rDNA. Another property of the vector is that it should be capable of replicating in the host cell. So vector should go and divide in the host cell. And vector should have a convenient restriction endonuclease sites for inserting the DNA of interest. As we have already discussed, restriction enzymes cut the DNA at particular site. So vector should have the convenient restriction endonuclease sites. And vector should be small and easy to isolate. Now let us discuss about vectors used in rDNA technology. Vectors used are BACS, that is bacterial artificial chromosomes, and plasmid. Apart from this, lambda phage vector, cosmid vector, expression vector, and YACS, yeast artificial chromosomes vector. Let us discuss about each vector in detail. Firstly, about the plasmid vector. Plasmids are nothing but the small extra chromosomal DNA molecules within a cell that is physically separated from the chromosomal DNA, and they can replicate independently. So basically plasmid is an extra chromosomal DNA which is physically separated from the chromosomal DNA. Plasmid can replicate independently of the bacterial chromosome, and it is useful for cloning the DNA inserts less than that of the 20 kilo base pairs. So please remember, plasmid is able to carry only the less number of base pairs. Next vector is the lambda phage vector. Lambda phage vector is nothing but a virus particle which includes the head, tail and tail fibers, and the head of that contains the double-stranded circular genome. All in all, these lambda phage vectors can convey the bigger DNA groupings than that of the plasmid vectors. As we have already studied, plasmid carry less than 20 kilobase pairs, but lambda phage vector can carry the bigger DNA segments. So that is the difference between the plasmid and the lambda phage. Now coming to the cosmid vector. What is a cosmid vector? Cosmids are the hybrids of the lambda phages and the plasmids. That means it has the properties of both lambda phage and the plasmids. So they can replicate like plasmids but can be packaged like the lambda phage. So cosmids are the hybrid vectors. Now coming to the expression vectors. Expression vectors are nothing but the plasmid or the virus which are used specially to introduce a particular gene into a target cell. A particular gene is introduced into a foreign cell, and these are useful for expressing the eukaryotic genes in the bacteria. Coming to the another vector, that is yeast artificial chromosomes. These are the yeast vectors that have been engineered, that means designed to contain a centromere, telomere, origin of replication and a selectable marker. So yeast vectors are designed to have all these components, and they are useful for cloning the eukaryotic genes that contain the introns. What are introns? Introns are nothing but the coding regions of the DNA, whereas exons are the non-coding regions. So these vectors clone the introns. Now coming to the bacterial artificial chromosomes. So what exactly is a bacterial artificial chromosome? It is an engineered DNA molecule. That means it's a designed DNA molecule used to clone DNA sequences in the bacterial cells. They're capable of carrying the DNA inserts larger than plasmids or the lambda phages. Plasmids carry less than 20 kilobase pairs, lambda phages carry little more than 20 kilobase pairs, and bacterial artificial chromosomes carry the larger DNA inserts. Coming to the techniques used in the rDNA technology. What are all the techniques used in recombinant DNA technology? They are gel electrophoresis, cloning libraries, restriction enzyme mapping, PCR, polymerase chain reaction, nucleic acid hybridization, and finally DNA microarrays. Let us now discuss about each and every technique. First of all, gel electrophoresis. It is a technique used to separate the DNA fragments according to their size. DNA samples are loaded into the wells at one end of the gel, and the electric current is applied to pull them through the gel. DNA fragments being negatively charged, they move towards the positive electrode. This is how the DNA fragments are separated. Now let us discuss about the cloning libraries. Cloning libraries are nothing but the collection of the DNA clones in a certain vector. Its main goal is to have each gene represented in the cloning library at least once. Then what is genomic cloning library? It is a collection of the total genomic DNA from a single organism. Then what is cDNA library? It is a collection of the cloned DNA sequences that are complementary to the mRNA that was extracted from the organism or the tissue. Next, coming to the PCR, polymerase chain reaction. It is a technique used to make as many as copies of the specific DNA region in vitro. In vitro means in a test tube rather than organism. Now coming to the restriction enzyme mapping. It is important to have a restriction enzyme site map of a cloned gene for further manipulation of the gene. As we already know, restriction enzymes, they cut the DNA at the specific site. So for this, restriction enzyme mapping is of utmost importance for further manipulation of the gene. Let us now know about the procedure of how the DNA fragments have been deduced. First of all, the fragments are subjected to the gel electrophoresis. What does this gel electrophoresis does? It will separate the fragments based on the size. So the restriction sites are deduced based on the size of the fragments. Next is about the nucleic acid hybridization. Under nucleic acid hybridization, we have southern blot, northern blot and western blot. Southern blot is a technique used to detect the specific DNA sequence in a sample. Northern blot is a classical way of analyzing the presence of specific RNA sequence in the sample. Southern blot is used for DNA sequence. Northern blot is used for RNA sequence. Please know this difference. What is western blot now? It is used to detect the specific proteins amongst the mixture of proteins. So western blot is used to detect the specific proteins. Coming to the final technique, that is DNA microarray. This is also called as DNA chip or the biochip. It is a collection of the microscopic DNA spots attached to the solid surface. This method is used to measure the expression levels of large numbers of genes simultaneously. So large number of genes at a time are evaluated by DNA microarray. Last but not the least, let us know about the applications of rDNA technology. rDNA technology is used in agriculture for growing the crops of your choice. It's also used in pharmacology for artificial insulin production. It's also used in gene therapy and also used in vaccination. rDNA is also used in growing the plants. So this is it about the topic recombinant DNA and biotechnology, and thank you.